So, in the previous couple of slides, we were discussing the fact that membrane composition is very heterogeneous, oftentimes, with respect to exactly what lipids are present in any particular membrane. So, that membranes are heterogeneous. Membrane composition also varies, as we said, between different kinds of membranes and different tissue types, for example. And we also saw that bilayer composition varies depending on which leaflet we're discussing. The point of all of those being, of course, that the chemical characteristics can be programmed. The chemical characteristics of the lipid membrane can be programmed by programming the composition of that membrane. In this slide, we begin to see what some of the results of the variable composition of the membrane are. And there's a couple of things that I'm trying to get across here. But overall, our title, of course, is Order and Disorder in the Lipid Bilayer. And first, let's just talk about what this cartoon shows. This is tracing the pathway that a lipid is taking using some fluorescent label in one leaflet of a bilayer over time. And we see that if we begin here, what we see is that there's a lot of motion. It looks more or less randomly bouncing around, but that we eventually migrate over to another region. And within that trajectory, there seem to be domains. And so this is very interesting. First of all, we see that the membrane can be considered to be fluid. That is to say, there are only non-covalent interactions holding a particular lipid in its position on the membrane, and therefore we should be able to move around laterally in a more or less random way. But within that randomness, there seems to be some organization, and perhaps that's because there are boundaries that are present, and those boundaries could be a matter of encountering a region of the membrane that has a slightly different lipid composition, or that has a slightly different protein composition, or perhaps there are other structures, usually that we're going to be talking about protein structures, but they might be peptidoglycan structures that also define domains on the surface of the membrane. So there's a dynamism to the membrane that depends on the composition of the membrane. One aspect of this is the possibility of the formation of rafts. So we might consider one of these domains of lipid organization to be a sort of a raft that's self-assembled based on, again, the affinity of the individual lipid components. And in this cartoon, there's a bunch of stuff going on, but I think it's easy to see that in this sort of outside region of the raft, we see that the lipid bilayer is shallower. That is, there's only a certain depth, and then we see that there's longer chain fatty acid groups on the membrane lipids that make this raft a little bit fatter, a little bit thicker. So again, those lipids self-assemble into these structures, and of course what could also affect the self-assembly is the presence of other lipids, lipids such as cholesterol, non-phosphoglyceride-based lipids, as well as proteins that are linked to the lipids bilayer through these prenyl groups or these fatty acyl chains, and the presence of other sort of membrane-associated proteins. So in any case, what we see is that membrane components segregate into microdomains and these microdomains sort of behave as lipid rafts. That is to say, they are still mobile structures, and they can move around more or less as a unit. A little bit more on the idea of lipid rafts. This is a nice cartoon that just shows some of the things that might affect lipid raft formation. Here's a raft phase, that is to say that it's the lipid composition and perhaps the membrane. Instead of lipids, let's just say membrane component composition that determines the structure of the raft. But there might be other protein structures that are extra to the membrane, such as some actin platform that can drive organization of the membrane as well. So there's some superstructures that define lipid composition as well, and might inhibit some of that mobility that we think of when we think of the fluid mosaic model of a lipid membrane. So, but as with everything, right, it's the chemical characteristics that determine the affinities. So the protein's affinities for the lipids in a particular raft structure, the lipid affinities for the other lipids in that raft structure, what proteins are present in the raft as well. So it's complicated, but you get the sense that within this idea of a fluid bilayer and a dynamic bilayer, there is organization that arises. Moving away from that subject, though, let's think about what happens to membranes as dynamic objects. And so this is a complicated figure. There's a lot going on here. Just giving you some of the different ideas or some of the different situations in which membranes have to fuse, right, join together with one another. Right, budding of Golgis of vesicles from the Golgi, or accepting of vesicles into the Golgi, exocytosis, so the merging of a vesicle with the surface membrane, so that we can eject stuff from the vesicle into the extracellular milieu. Endocytosis, the opposite, obviously. We've talked already in previous lecture this week about viral infection and how that requires the merger as mediated by hemagglutinin of the viral membrane with the cell membrane, the host membrane. Fusion of egg and sperm, obviously, fusion of vesicles, etc. And then cell division, of course, is another obvious one. So there's many places where we have to think about what kind of process is going on that might drive the merger or separation of membrane structures. So membranes are constantly being added to, taken away from, and recycled. They're constantly changing. And frequently we will see that membrane fusion is mediated by specific fusion proteins. Now, we talked about hemagglutinin bringing two membranes into contact with one another, and the chemical changes in hemagglutinin structure allow for the merger of those two membranes. But there are plenty of other systems that organize these membrane fusion events. And a really nice one that at least originated in the description of fusion of vesicles and neurotransmitter release are the SNAP-SNARE complexes, and SNAP just stands for soluble N-ethylmaleimide sensitive attachment protein and the receptors for those proteins. And I'm not showing you a cartoon of this, but basically the point is simply that these are two proteins that are membrane-bound or membrane-associated proteins that have an affinity for one another that drives the merger of the membranes to which they are attached through bringing those membranes into proximity, similar to the hemagglutinin story, although not carbohydrate mediated in this case. So in terms of membrane fusion, right, or trafficking of stuff throughout the cell, receptor mediated exocytosis. Here is the story, and this is just a sort of a cartoon. It's not actually a time lapse, but you can see the different steps in the process of this endocytosis event, and this is via a clathrin-coated clathrin-coated pit. So clathrin being a protein that's associated with a membrane, and that clathrin migrates to a region or a domain of the membrane based on the activation of a particular kind of receptor. Again, I'm skipping the cartoon here, just giving you the overall story. When the clathrin migrates to a particular domain of the membrane, that affects the structure of the membrane, right? And we see that happening as this begins to take on this bowl or invagination, and we see that very clearly here. And eventually, we essentially the clathrin is creating a structure around which the membrane must assemble itself, and so we'll eventually pinch off here, and we see this vesicle that's been brought into the cell. Similar disassembly is required for an exocytosis event through a clathrin-mediated process. So there are lots of proteins associated with transit across the membrane or fusion of membranes. Let's think about some different kinds of proteins now, not merger of membranes, but movement of stuff into and out of and across membranes. So this is just a sort of a general overview of the possible kinds of transporters that one might encounter. There are passive transporters that simply move compounds along their gradient, along some chemical gradient. That is to say, if it's a uniporter, it's the simplest one to imagine. There's a molecule, a solute molecule in the extracellular environment that's at a high concentration. The uniporter moves it down its concentration gradient to try to reach an equilibrium across the membrane, and so the solute flows into the cell. A symporter would be a molecule that uses a concentration gradient of one solute to move something else, a second solute, into a membrane. And an antiporter is similar in that we use a chemical gradient of one compound to move another compound in the opposite direction. So those are passive transports that use essentially the energy stored up in chemical disequilibrium across the membrane. Now, active transporters are another kind of transporter that use a chemical energy to move a solute, and that's often going to be ATP hydrolysis, but not strictly so. And then there's simple channels. And this determines a membrane's solute permeability, so we could imagine that the membrane would be permeable to different kinds of solutes depending on what kind of channels there are that might selectively allow transit of one kind of compound and not another. There are many channels that we could be discussed, but here's two examples: the aquaporins that regulate osmotic pressure by allowing the flow of water, and then of course ion channels that regulate electrical potential across the cell membrane. Okay, so looking a little bit more closely at a couple of different examples of transporters. This is a potassium channel, and the interesting thing about this channel is that it's very specific for potassium ion. So not only does it like positive charges, but it prefers a smaller cations and rejects larger ones. And we can think about a structural basis for this. The cool thing about this story is that the backbone carbonyls of the interior of this channel, so presented, let's say that again, the carbonyls presented from the backbone to the interior of this channel of the transmembrane protein replace the hydration shell that typically solubilizes or solvates the potassium ion, and that creates a selectivity filter. So we're only going to admit an ion whose solvation sphere can be replicated by the interior surface of this channel, if that makes sense. And in a different example, we can gate an ion channel by using a conformational change induced by some other ligand binding to drive the opening of the channel. Now that can be a ligand gating mechanism, or it can also be a voltage gated mechanism, and we're not going to talk about the difference of those, but the key notion here being that the channel itself can act as a sensor. Remembering that what has to happen in the channel is the transport of an ion across the membrane, and we can respond either to a concentration change in some other ligand, say a neurotransmitter, or say in some voltage gradient across the membrane. So, but let's go back to this potassium channel. No, let's skip it. I think that's good enough for now. But here I think the point was that we see this desolvated ion, right? This desolvated ion. Here's the solvated ion, and it enters the channel. We have this sort of trap here for this guy. That's the interaction driven by what's this? Helical dipole. We've seen this story before, but there's a desolvation event that occurs, and then the ion can then enter the channel as if it were still solvated by water. Of course, it's not. It's solvated by, again, those backbone carbonyls. Okay, another example of a transporter. This is a multi-drug resistance antiporter example. So these are antiporters that are involved in the efflux of drugs and other xenobiotics. And by xenobiotic, I just mean small molecules that might be considered toxins, anything foreign in the organism. Your liver especially has very... and kidneys and other, actually many tissues have these channels, but the point is that we have a complicated system in place for the rejection of foreign solutes from the system. And this is just a cartoon of that molecule, one of those molecules, and you can see that it's kind of standard configuration in that it's this big helical structure that's got a large nonpolar region that interacts with the lipid bilayer, and there's polar regions at the extremities as we emerge from the bilayer for exposure to solvent. There's a conformational change that is critical to this once again, and this is, again, it's a notion that we've seen many times. That is to say, a ligand binding event drives a conformational change, and that results in the activation of the protein. In this case, the ligand binding event is the binding of this rubidium ion, and you can see that there's this interesting binding site for it composed of something that's maybe other than the usual suspects, but certainly at least there's a glutamic acid and a tyrosine involved in immobilizing this guy. The key, however, is that there's a large cavity on one surface of the membrane, on one surface of the protein, on one face of the membrane, that can admit the ions, right? And then what happens is we bind our ion in that cavity, and we see that you might even be considering that there's a particular negative charge density that might essentially suck our ligand into to this cavity, and then that binding event induces a conformational change, which opens a cavity on the opposite face of the membrane, and the toxin molecule can then be admitted. And this is an antiporter, remember, so the ligand is coming, the rubidium ion comes in, flip in the conformation, rubidium ion goes out and is exchanged with a toxin molecule, and then the toxin can then leave and exit into either to be dissolved into the membrane, depending on what kind of structure it is, or released into the exterior of that compartment. So, but again, this story isn't anything new really either. It's just that the conformational change that's coupled to this ligand binding event results in the presentation of a new binding site for the molecule, the drug that we are trying to develop a resistance to. We develop a new binding site for the drug, which binds and then is released into the extracellular environment. Okay, so we've talked about membrane structure and dynamism. We've talked about merger of membranes. We've talked about how proteins can help facilitate transfer of stuff across the membranes. Let's also think about the transfer of information across the membranes. And we've talked about signal transduction in other contexts in this course, but for our last lecture this semester, we're going to review some of that information one more time. This is a sort of a generalized cartoon of signal transduction. Here's a membrane. This little bar here is supposed to represent a membrane, and there might be several proteins associated with the membrane. But the membrane, of course, is where we have our receptor, transmembrane receptor bound, and that receptor responds to the presentation of some external stimulus. It could be a variety of things. We've talked about voltage being a potential signal. We've talked about neurotransmitters being a potential signal. We've talked about rubidium ions being a particular signal just today, but of course there are many hormonal signals, and we'll come to talk about those in the next couple of slides. In any case, there's something that happens at the membrane receptor, and often that's a conformational change. It might also be a post-translational modification like methylation or acetylation, but whatever it is, that change induces a change in a downstream molecule. We'll call this a transducer molecule. So this molecule senses the change in the receptor state and then transfers that information to an enzyme that affects the change, that affects the development of high concentrations of second messengers that then go on to mediate the downstream effects of the signal transduction cascade. Now, the principal second messenger we've talked about in this class is cyclic AMP. We've talked about others as well, but let's just stick with that paradigm for now. And just remember that we have to respond to an external signal through a conformational change at the membrane receptor that results in the activation of an enzyme that generates a second messenger on the interior of the membrane. So what's been transferred is not stuff, physical solutes across the membrane, but information in the form of conformational change. Surprise, surprise, surprise. Okay, so the example that I'm building up to is that of the G-protein coupled receptors. Those involve a severin transmembrane alpha helical receptor molecule. Here it is. There's a nice rainbow colors for your alpha helices embedded in the membrane. And of course, on one face of the membrane, we have a binding site. And here you can see whatever this ligand is. I'm not even sure I remember exactly what this receptor is. It doesn't matter, but there's a binding site. And of course, when you insert your ligand into the core of this helical bundle, there's a configurational change that occurs. That is manifested, as I say here, on the cell interior. The key change is that conformational change is coupled to the behavior of a heterotrimeric G protein in this particular sort of archetypal example. So G proteins are often these heterotrimeric molecules. We have an alpha subunit that is a GTPase and a beta gamma subunit that are often interacting with each other. Sometimes they separate as well. In any case, the heterotrimer interacts with the receptor in its unactivated state typically, although we might see the opposite being the case as well. But upon receptor activation, the heterotrimer dissociates from the receptor and dissociates into at least the alpha subunit and the beta gamma subunit. And then the alpha subunit becomes the transducer in our sort of archetypal example, going on to activate a downstream effector enzyme. A key here is that I'm mentioning is that there are several genes for each subunit, so what I want you to imagine is that I'm presenting you with this very simple picture of G protein interacting or mediating the information flow between the receptor and the effector enzyme. But what we have to imagine is that there's a very complicated host of heterotrimeric G proteins that are tuned to interact with a very complicated set of receptors. So we can attune the cell or tune the G protein to react with an individual signal transduction pathways or multiple signal transduction pathways. Okay, so as I say, receptor binding usually results in the dissociation of the trimer and activation of the alpha subunit. Here's the alpha subunit. The monomer now is free to bind with other partners. That's critical because it's going to go on and interact with other partners and activate them. The key, however, as I've mentioned it in several previous lectures, is that this is a self-timed process. Right? This is a GTPase, and the degradation of GTP to GDP results in the deactivation of the G protein. So as long as GTP is bound, the molecule is active. But when it hydrolyzes GTP, it's ready to reassociate with beta gamma and the receptor again. So this is a sort of a built-in timer for how long the system responds to that initial stimulus. Now, of course, there are other molecules that we're not going to go into this in any great depth, but there are other molecules that might mediate the rate of hydrolysis, so it allow us to attenuate the signal or lengthen the duration of the signal. So there's always those extra complications. And in this cartoon, this is just a cartoon showing you what those conformational changes are between the release of GDP and the binding of GTP that happens concomitant with receptor activation. And of course, there's the usual stuff that happens, big conformational changes, blah, blah, blah. In this case, next slide, coming to the end here, G-alpha interacts with multiple targets, could interact with multiple targets, so we could, again, the idea is that we amplify that initial small signal many fold. And here's the GTP form of this alpha subunit of a particular G protein interacting with adenylate cyclase. Now this is just, again, adenylate cyclase is another membrane protein, but we're looking at the soluble fragment of that. And of course, obviously, we've discussed before, adenylate cyclase takes ATP and converts it to cyclic AMP, and here's that cyclic form. And this molecule then goes on to act as a second messenger intracellularly. I'm not going to dwell on this slide very much. We've already discussed the fact that these enzymes are often organized by placement of regulatory subunit with a catalytic subunit, and in this case, this is no different. We have a regulatory subunit. This is, again, downstream of adenylate cyclase. We have some kinase that responds to the presence of cyclic AMP through allosteric effects at the regulatory subunit, and binding of regulatory subunit by the second messenger results in the release of those active catalytic subunits, which go on to, again, the kinases that are activated now go on to activate other downstream members of the signal transduction pathway. Again, this is just a canonical picture of the adenylyl cyclase pathway. This is a hormone-activated pathway. You see that we might begin with a stimulatory hormone or an inhibitory hormone, depending on exactly what we are trying to accomplish. And exactly what we do accomplish depends on the molecules that are activated downstream of the stimulation event. But in any case, we have the stimulatory G protein that interacts with adenylate cyclase, generates cyclic AMP, and blah, 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 we have a cellular response. The same thing might happen through an inhibitory hormone. So again, what happens after receptor activation depends on the composition of the players in a particular system. And insulin signaling is a great example of this. It's another standard example of this. We have a sort of a complicated receptor structure in which the kinase, right, is not adenylate kinase, but there's a kinase that's integral to the receptor itself. So we have this large structure. We receive our signal at insulin, at the receptor via insulin, and that induces a conformational change that activates the protein tyrosine kinases. So there's an autophosphorylation event, and so there's a phosphorylation of the receptor itself that drives its own activation, and then once phosphorylated, of course, the receptor kinase is free to engage with a number of other enzymes or a number of other molecules, and the molecules that might be available for phosphorylation sort of determine what the ultimate downstream signal is. Okay, now my last slide here, I guess the last slide of the semester, is this incredibly complicated insulin signaling pathway. And I'm not even going to delve into this. We might discuss it in class just for fun, but the bottom line is that there's, upon receptor activation, binding of insulin at the receptor, there's a whole slew of events that take place. And I guess the key with this slide is that many responses result in gene transcriptional changes, the biosynthesis of new proteins, but others are changes in enzymatic activity. We'll go into these details in class. Of course, remembering that what the main job of insulin seems to be the presentation of new glucose transporter molecules. So there's our transporters rearing their head again at the cell surface. So insulin responding to an increase in sugar in the blood, that response needs to be presentation of transporter molecules that allow the transport of the glucose into the interior of the cell. And you see that, again, I'm not going to go into them here, but there are a variety of events that are taking place, some of which are excitatory, some of which are inhibitory, and the main point of showing you this slide, however, is to give you a sense of the complexity of those signal transduction pathways and everything that happens downstream of that receptor activation. Okay. Hope you enjoyed it. See you in class.